Hey guys, this is Miss Hunt. I'm going to try to get my Elmo working so we can do some Elmo. Let's see. I don't want to record. I just want to have it open. Okay. All right, this is Miss Hunt, and I'm going to start my video now. So this is Miss Hunt, and what we're going to do is, after yesterday, I had a lot of kids say, I am lost. So this video is for my lost kids. All right, what I recommend you getting, I'm going to have some colors here to help us out. I'm also going to be using some string to help us out. And we're going to have, of course, some pens because color is good. Our eyes like color a lot. So we're going to zoom out here. Okay. All right. So I'm kind of, let me move my camera here so you can see me over here too. Okay. All right. So a little review to get us started. In our previous unit, we talked about DNA, okay? And we talked about with DNA, we talked about DNA replication. And we also talked about protein synthesis. All right, with DNA replication, if you remember, there was a couple of enzymes who were in charge with this. We had helicase, and his job was to unzip or unwind the DNA. We had RNA primer or RNA primase. Okay, and his job was to lay down the piece of RNA so that the DNA knew where to begin. We had the DNA polymerase, who was the builder, who built the new nucleotides. And then we remember that he worked going towards that 3' direction. And there was the leading and lagging sides. And on the lagging side, he had to work backwards. And we ended up with these fragments called Okazaki fragments. And those fragments had to be glued back with ligase. Okay. So that's what we did with replication. We ended up with 2 new strands that were half old and half new. And remember, we called that semi-conservative replication. Remember, half of it was the old strand and the other half was new. Okay. And we said that they did that so that you had less of a chance of having mistakes and messing up. Okay. All right. So that was DNA replication. The other part was protein synthesis. In protein synthesis, remember you had transcription, in which the DNA went to make the mRNA. Remember the mRNA was just one piece of RNA like that had the codons on it. Remember this left the nucleus. So this was the nucleus. And it travels to a ribosome. And then we had translation, in which that mRNA came in, and we had those big tRNAs bringing the anticodons, and we built proteins. Okay, so that was last unit. It's important in this unit, though, that we're studying, because this right here, the DNA replication, is going to play a role. Okay, so I might have to pull this back out in just a minute. All right. So as for the new stuff, we're studying the cell cycle, which means I'm going to make a new cell. And in order to make a new cell, I need a new copy of that DNA to go in my cell. So we kind of reference back to DNA replication as we're talking about stuff like, oh yeah, if I got to make a new cell, in that new cell's got to be a copy of the DNA. So let's go through real quick the cell. Okay, here's my cell. We're going to make a little animal cell. So we're drawing a circle. Remember, I have a nucleus. He's a dark area. Okay, and there's all those organelles inside, the ER, the mitochondria, the lysosomes, the Golgi body. Everybody's in there doing their job. Okay, all right, so I need to make a new cell. And what happens is I'm going to end up with two identical cells, okay, after the cell cycle. Now, why? Why make new cells? Our notes talked about that if cells got too big, they were not efficient. Okay, they had a hard time communicating. They had a hard time transporting. And small cells are better. So as a cell grows and it gets bigger and bigger, the best thing to do is split and make two new cells, because small cells are better. So you remember we did the diffusion lab with the agar cubes. We put those little the dark purple or dark green cubes in vinegar, and they turned bright orange. And we saw that the vinegar diffused through those cubes. The small cubes, it diffused really quickly, but the big cubes, it took a long time. So let's think back real quick on protein synthesis. On protein synthesis, the mRNA leaves the nucleus and it travels to a ribosome. If my cell was really, really big, this distance would be really far, and it might get lost, it might get damaged, there might be problems. After the protein is built, the protein gets packaged by the ER and sent to the Golgi, and the Golgi sends it out of the cell. And again, if my cell was too big, the protein could get lost. Something could get damaged in the process. So it's really nice to have a small cell so that we can transport proteins and material really quickly, and we can communicate within the cell really quickly. So we need to have a new cell because small cells are better. Are we okay with that? All right. Now, let's talk about this cell cycle. How do we make a new cell? My favorite purple pen seems to be drying out, which makes me sad. Okay, let's get a different pen up here. Okay. Ooh, I found another purple pen. Okay, let's try this one. So, in order to build a new cell, first of all, we have to memorize the steps in order, and then we have to know like what they are, what they do. Okay, so here is the cell cycle. First, there are 3 phases to the cell cycle. The first one is interphase. All right, the second part is mitosis. And the third part is cytokinesis. Okay. Ooh, might not be able to see that blue. Okay. All right, so these are the three parts of the cell cycle: interphase, mitosis, and cytokinesis. Interphase, mitosis, and cytokinesis. All right, interphase is the longest. All right, and this phase is actually split into three phases. It is G1, S, and G2. Okay, and yes, you have to know what each one does. So in G1, the cell grows and does its job. So what's its job? Building proteins. Okay, its job is also to do cellular respiration, if it's a plant, to do photosynthesis, to take in nutrients through the cell membrane. All those things are going on. But basically, it's just doing its job here. Remember, this is where it spends most of its time. In the S phase, we're synthesizing DNA, okay? DNA synthesis is happening here. So what does that mean? Right here, we're replicating our DNA. The helicase unzips, the RNA primase primes, the DNA polymerase builds, and the ligase glues the Okazaki, and you're left with a semi-conservative replication DNA. This happens in the S phase right here, just a small part of the whole phase of interphase. All right, G2. We prepare for nuclear division or mitosis. So we're getting ready and preparing. All right, mitosis is the division of the nucleus. Okay, division of the nucleus. So this is the cells growing. So let's do our cell, and our cell grows a little bigger. Okay. And remember up here, it can't keep getting bigger. It's got to split apart. So we're growing our DNA copies, and we're getting ready to divide our nucleus so that we can make two cells. All right, mitosis is split into 4 stages. Okay, 1, 2, 3, 4. This is prophase, metaphase, anaphase, and telophase. Okay, we remember it as PMAT. PMAT is mitosis. Mitosis is PMAT. PMAT is mitosis. Mitosis is PMAT. All right, prophase, metaphase, anaphase, and telophase. And I'm going to be drawing pictures of all of these phases to help you out to understand what happens. Okay? After the nucleus divides, so here we have the nucleus is going to split apart and it's going to be making a new one. Okay? Here we have cytokinesis, which is when the cell splits into 2 daughter cells. And they both have a nucleus. I should be drawing the nucleus and all these. Okay, that looks identical to that parent cell. Okay, we call these 2 cells daughter cells. Daughter cells. And they're just replacing the original cell. Okay, we're just making a new cell and replacing the original cell. So at this point, you should have the phases in order. Okay. All right, so in class, we're going to do this worksheet, okay? And we're going to put the phases in order. So if right now you want to pause the video and pull that sheet out and see if you can do it right, that's great. All right, and then let's check our answers to see if you did it right, okay? All right, so we should have interphase, interphase, and interphase. And we should have G1, S, and G2. Okay. Now, the only thing I didn't tell you on the other sheet, this is called the cell division. It's also called the M phase. So let me go back and put that on there. These two, mitosis and cytokinesis, are also called the M phase of cell division. Okay? All right. So, move that over there. Okay. Let's go through it again. We grow, we synthesize the DNA, we prepare for nuclear division, and then we have prophase, metaphase, anaphase, telophase, cytokinesis. And remember, PMAT is mitosis. Mitosis is PMAT. Okay? Hopefully, you got them in order. I know you don't know what mitosis looks like, but I hope so far we're doing okay. All right, flip the worksheet over. And again, if you want to pause the video, that's great. And then you can try it out. And then we can go over our answers. Okay, and you can see how bad my handwriting is. Okay, all right, here are the answers for the questions, and that way you can look up and see if you're doing it correctly. This is basically, did you get them in order? Okay, do you have the same things as I do? Are you understanding what happens in each of the phases? Okay, all right. So I am going to go on to the next thing. Okay, all right. Let's draw out these phases and see if we can get the phases. Okay, all right. Before we do the phases, we've got to go through some vocab words. Okay, let's go through some vocab words first. Okay, sorry about that. Okay, chromatin. All right, chromosome, chromatin, centromere, homologous chromosomes, and sister chromatids. Okay, guys, it's about to get real. How many C-letter words can we squeeze into cellular biology? I know it's very difficult. And believe me, it's frustrating as a teacher to try to teach it. Okay, so do you remember the big blue poster that had all the different traits on each of the chromosomes that we had? My kids thought it was funny. We looked it up, and there was maple syrup urine syndrome was one of the syndromes that the kids saw. They thought it was very bizarre. Red hair was on there. Eye color was on there. All sorts of things are on your DNA. And where's your DNA? It's in the nucleus of every cell. Okay, so let's go through these words real quick and try to understand what we're talking about. Okay. All right. You have 46 chromosomes. Every species has a different number of chromosomes, and I don't know any of them off the top of my head. Monkeys, I don't know, but bananas, all these different things, everything, fruit flies, they all have different numbers of chromosomes. And it does not correspond to how complex you are. It's not like humans have 46 and a plant has 4. No, some fungi have 100 different chromosomes. So it doesn't matter to how complex of a species it is. It's just a different number of chromosomes in every species. Humans have 46 chromosomes. We say 23 pairs of chromosomes. And this is actually 23 pairs of homologous chromosomes. There's a Latin there, homo. You've heard of homo before from homogeneous mixtures and homozygous traits. Homo in Latin means same. So on that poster, we looked at 23 pairs of chromosomes. So let's figure out what that means. So we have chromosome number 1. Let's just say we had chromosome pair number 2, and then number 3. And this goes all the way 4, 5, 6, 7, 8, 9, 10, 11, 12. We have 22, and then we have 23. Okay? You inherited a chromosome from your mom, from all of these. And you also inherited a chromosome from your dad, from all of these. Okay, so in total, this is 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. I could go all the way, and that would be 46 chromosomes or 23 pairs of chromosomes. When my nucleus needs to make a copy, the DNA in all of this needs to be the same. I need my new cell to also have 46 chromosomes, 23 pairs of the homologous chromosomes. So these right here are homologous. Which means the same trait here from mom is the same trait here from dad. Okay, so let's just do hair color. Okay, which one will you express? That's genetics. Whether you express the dominant or the recessive, or if it's a blend of the two traits, that's genetics, which we'll be covering after Christmas. But right now, you just need to know that you're inheriting one from mom and one from dad, and your new cell also needs to have this copy of every bit of information of you in every nucleus of every cell. Okay? All right, so I hope by seeing pink as your mom and blue as your dad, you can see that they're coding for the same thing, but the actual traits might be a little bit different of what you get. But your new cell needs to have both of these in it. Okay? So let's focus on just this first one, just chromosome pair number 1. Okay? So as we do our drawings and everything, we're just going to have just this one. Okay? So remember, we're looking at chromosome pair number 1. Okay? So we start off with an original, one from mom and one from dad. Okay? And we'll call this the parental DNA. All right? In the cell cycle, G1, remember this, G1, my cell grows. But in the S phase, I need to make new DNA. So let's do the S phase. In the S phase, I'm going to double my DNA. So I'm going to have a whole chromosome, and I'm going to pair it up with a whole chromosome. And then I'm going to double this guy. And if you notice, I'm kind of drawing it kind of funky, okay? I'm not really drawing it as an X. So let me show you on this paper right here, okay? I'm not doing crisscross, okay? That's not what I want to do. Instead, I want to draw it like this, like a boomerang, and then here another boomerang, okay? Sometimes you don't even have to make them touch. We can just go like this because I want to show that there are two separate guys here. And what they're going to be doing is be held together in the middle with this black guy named a centromere. So again, let me draw it. It's like two little boomerangs. Don't draw an X, even though they look like X's. It's just going to get you confused. Okay, so I'm going to put my little black dot in the middle here because that guy is a centromere. And the centromere holds these guys together. Now, what are their names? Oh my goodness. All right, this is called sister chromatids. Okay. These were called homologous chromosomes. Because they are the same information, but one from mom and one from dad. When they double, we call them sister chromatids. Okay, so here's another sister chromatid. We have other names for them. Okay, each one is called a chromatid. Chromatid. So this is two chromatids here. That's why they're sister chromatids. So this is a chromatid, that one strand, and then this is another chromatid. We also call them one chromosome. I know this is kind of crazy. You're like, but there's two of them. Yes, I know. When we count chromosomes, we only count centromeres. So we say 1, 2. Up over here, these guys also have centromeres. So this would be one chromosome, one chromosome. So these are two chromosomes because that gives us our 46 chromosomes. But this is considered one chromosome because the centromere is holding two chromatids together. This is considered one chromosome because the centromere is holding two chromatids together. They are sister chromatids. These two are homologous chromosomes because they're the same information. Okay? I know it's kind of nutty. Okay? All right, so that gives us a couple of good vocab words. Okay? I didn't cover chromatin. That DNA, you remember it winds up around those histones and those ketones. As it becomes a little bit more condensed, it becomes chromatin. And as it winds up really, 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 really tightly, it becomes a chromosome. So the chromatin is not a chromatid. It's just the loosely piece of DNA that before it becomes a chromosome. And remember, this is one chromosome. This is one chromosome because we count the centromere. These are two chromatids, chromatid, and they're sister chromatids. Okay? All right, and I know that is difficult. okay? But hopefully with repetition, we're going to get to understand this. Okay, so basically what happens in my cell with my nucleus, okay? This is after G2, okay? My cell, the nucleus starts to disappear, and these little guys start to form called centrioles. We say centrioles at the poles, and there's 2 centrioles here. The nucleus starts to disappear. Okay? We get the chromatin is loose. I say it looks like a bowl full of noodles. Okay? I should put the centrioles here. Okay? The sister chromatids start to find each other. Centrioles at the poles still. And something starts to come out. This is called spindle fibers. They start to form. So the sister chromatids pair. Okay? Excuse me. All of this is prophase. All of it. That's prophase. Okay? The nucleus disappears. The centrioles form. The chromatin is out and it starts to condense. The sister chromatids find each other and the spindle fibers start to come out of the centrioles. They're going to attach to these sister chromatids, and they're going to be moving them into the middle. Okay? Metaphase begins. I draw it a little bit bigger for us. Okay? Centrioles at the poles. What we want is these sister chromatids to begin to line up in the middle. Okay? Spindle fibers are pushing them there and putting them in the middle. They're attaching to the centromeres in the middle. Centromeres in the middle. So we say metaphase middle, it makes us look like they're in the middle of the cell. Okay? Metaphase in the middle. All right? Okay. Then we go to anaphase. Now you see, we're in prophase for a lot. There's a lot of stuff that happens in prophase. Metaphase is just they're in the middle. That's it. And they are, the sister chromatids are in the middle. Okay? Anaphase, we say ana away. Anaphase away. Okay? We have what I think look like flying V's. Here we go. Okay. And I really should get rid of one of these down here or one up here. Let me get rid of this guy. Okay. There we go. So I begin with 4. 1, 2, 3, 4. And yeah, I should begin with, because they're finding each other and pairing up. Okay. So they've already doubled. So I really began with, here, I really began with 2. So my new one should end with 2. So I'm going to do a little line here and say, in that nucleus, we started with 2, okay? Now, in a human, we would have started with 46. We would have gone up to 92 here because everything would have doubled, okay? 46 chromosomes, but to make my picture easier, I'm just using 2, okay? All right, anaphase away, what's happening? The sister chromatids have been broken apart, okay? And they're being pulled to the poles. Anaphase away, okay? And then we do telophase. In telophase, we start to get pinching happening, okay? And we start to, centrioles are here. They're still holding on to my little chromosomes, okay? A nucleus starts to form again. So here's my nucleus starting to form in telophase, okay? What's the phase after telophase? It's not a part of mitosis. It's cytokinesis. That was the last phase of the cell cycle. And just for our records, we're going to draw it like this with our nucleus, and we're going to put 2 in here so that they're identical to my parent cell, my original. And these are called daughter. Okay? All right. So we're left with 2 cells. And who does mitosis? Okay? All of your body cells. We also call those somatic. So my kidney, my hair, my eyes, my liver, I want every cell to be replaced with the exact copy of my cell. So that means they're going to do mitosis. And if you understand everything in this video, then you are doing really, really good. Okay? All right, I will have pictures of all of this up on the Google Classroom, and hopefully we can figure it all out. We are going to go over it again. What we're going to do is watch the Amoeba Sisters video tomorrow, and that should help us go over everything again. And you can read the book, watch the videos. All that is really good to help go over, and I hope that helps.